So, this is the live interactive session for the third week, and we have already had interactive sessions for the last two previous weeks. So, I'm not sure how many of you have attended the last 2 week sessions. And first, I'll ask that is there anyone who has not attended the last 2 weeks sessions? You can write down yes or no in the chat box. Is there anyone who is joining this interactive session for the first time? Please don't raise hands. Like if you have any queries, please write down in the chat box. Okay, okay. Yes, ma'am. For raised hands. Is it? Okay, yes, ma'am. Third class. Yeah, I want to know if is there anyone who is new to the class? Like, is it? It's their first class. Is there anyone who is joined for the first time? Yes, ma'am. I'm first class. Okay, okay. To all those who are for the first time, I welcome you all. I'm Amrita, and we'll be having this interactive session so that you can revise. Uh, so that you can revise your last week's course, whatever has been taught through the lectures by Professor Manak Das. So I hope all of you have been following the lectures. Every time I request in the class that before you uh, be a part of this interactive session, it is very necessary that you attend the lecture of this week, right? We'll be discussing questions here, and I'll give you time to answer those questions. So only if you have attended the lectures, you will be able to answer those questions well, right? So this is an interactive session. So I'll be asking you questions first, and I'll give you 1 minute time to think and answer the questions. You can answer your options, your choices in the chat box, and after that, I'll tell you what is the right answer along with the correct explanation to that answer. Okay. So this is how we'll proceed with the session. Okay. So okay, it's already 6:15. Uh, Okay, whoever is new to the class, I want to know from them that are they in their bachelor's, like they are doing BSc, MSc, or some other course or PhD. I want to know that. Whoever is new to the class, I want to know from them. So life sciences. Okay, okay, yeah. Yeah, one more thing I want to say you all is if you have any queries regarding this interactive session, there is a discussion forum at the NPTEL portal. You can go there and write down your doubts or queries, whatever you have from this interactive session, you can write down there, and I'll be checking that discussion forum, and I'll and answer your questions or queries if you have any. Okay. And I want everyone to be active throughout this class. Answer actively. Doesn't matter even if it's wrong or right. It's okay if you have not attended the lecture and if you're guessing the answer right now, it's okay. But try to guess it well and try to answer it. Like write down in the chat box. Don't stay silent. I won't be able to know if you don't answer it also, right? But I'll request everyone if you have joined the class, answer it wrong or right. It's okay. We'll discuss it after that. Even if it's wrong, you'll know the right answer. Okay. Okay. We'll start now. It's already 6:15. Okay, is everyone able to listen me properly? Like, is the audio clear? As well as, are you able to see my first slide? Okay, okay, great. So we'll start with the first question, and I'll give you 1 minute time. Think wisely and write down in the chat box. After that, we'll discuss the answer to it. Okay. So your first question is, Which of these is not a muscle type? Which of these is not a muscle type? Skeletal muscle, cardiac muscle, smooth muscle, or rough muscle? Okay, and your time starts now. Start answering quickly. Okay, rough muscle. Okay. Okay, I need answers from everyone, whoever has joined the class. 60 people are here, so I want 60 answers. Which of these is not a muscle type? Okay, not. Skeletal muscle, cardiac muscle, smooth muscle, or rough muscle. Okay, I guess most of you have given the answer as rough muscle. Let's see if that kind of muscle exists or not. So, the correct answer is rough muscle. Congratulations who have given the right answer. And yes, one more thing I want everyone to please note down, like how many correct answers you're giving, how many wrong answers you're giving, and I'll ask in the end of the session, like what is your score out of 10, right? Or maybe 9. Today we have 9 questions. So the correct answer is rough muscle. So, what is the explanation, right? There are only three kinds of muscles we have in our body, that is, cardiac muscles, skeletal muscles, and smooth muscles. There is nothing like rough muscles. Skeletal muscles are found around the bones in our body, cardiac muscles are present in our heart, and smooth muscle cells are present in some organs like kidney, intestine, etc. Right, we'll know about these muscle types in detail in the upcoming questions. Okay, so this was a very simple question, like whatever rough, there is nothing like rough muscles. Okay, and rest three are the types of muscles. If you have attended the lectures, you must have known about it, right? So we'll move on to the next question. So the second question is, which of these muscle types is responsible for peristalsis in the esophagus? First option, skeletal muscles, cardiac muscles, smooth muscles, or rough muscles. Which of these muscle types is responsible for peristalsis in the esophagus? Your time starts now. Start answering. Whoever are raising hands, may I know what is the problem? Smooth muscles. Okay, most of you are saying smooth muscles. Is everyone giving the answers? I want 63 students are here. I want 63 answers. Which of these muscle types is responsible for peristalsis in the esophagus? Whoever are giving the answers, try to think what is the, like, what can be the correct explanation to it. I'll ask that also. Okay, should we move on to know what is the right answer? Most of you, I think everyone has given the answer is smooth muscles, and yes, it is the correct answer. Smooth muscle is responsible for peristalsis in the esophagus. So first of all, I want to know from everyone, what is peristalsis, right? You must have already learned in your class. I want to know what is peristalsis. Please write down in the chat box. Can anyone define peristalsis? Like, it's okay, even if it's brief in your own words. I just want to know if it is clear to everyone what is peristalsis. The automatic wave-like movement of muscles that line your gastrointestinal, the type of muscle that peristalsis, which is food, kind of movement that takes place at the food pipe. Okay. Good, good, good. I guess you all are attending the lectures. That's really nice. Okay, so peristalsis is a series of wave-like muscle contraction which moves food along the digestive tract, right? So it is basically a movement, contractions, muscle contractions, that is smooth muscle contractions which help the food, like the bolus, to go along the digestive tract for digestion, right? So here you can see the picture. So the food we have eaten after it went, if after it goes on to our food pipe by the salivary glands by mixing with the saliva, it is now called as bolus. And so the through the bolus is moved throughout our digestive tract through the muscular contractions, that is the smooth muscle contractions. The smooth muscles are present in the esophagus, and while they contract, it helps the bolus, it forces the bolus to move forward, right? And the waves of these muscle contractions move the bolus to move towards the stomach, right, where the digestion takes place. Okay, part of digestion takes place. Okay, this was a very simple question. We'll move on to the next question. Which of these is not true about myotubes? Which of these is not true about myotubes? Mind it, not true. Myotubes join to form muscle fiber. Multiple myotubes join to form myocytes. Myotubes are multinucleated. Myotubes are electrically active. Okay, the time starts now. Think wisely and answer. Okay, so saying C should be okay. Through these interactive sessions, we'll discuss one concept through one question, right? So which will help you to revise the lecture, and you will be able to solve the present year assignments quickly, right? You all must have been given a present year assignment, like 10 questions, and these questions will help you to answer those questions as well, right? Main motive is that your concepts should be clear. Okay, I'm guessing everyone has not given the answer yet. Please try to answer, it's okay. Even if you don't know, try to guess it. Okay, many are saying B, many are saying D. Lot of confusion is there. B or D. Okay, I guess everyone has answered. So the correct answer is B. Okay, good job everyone who has answered, the ones who have answered B, and those who have answered D is the wrong answer. Myotubes are electrically active. It is a correct statement. A, C, D, all of them are correct statements, and only which is a false statement is multiple myotubes join to form myocytes, right? So we'll know how. So first of all, I want to know what are myotubes. Can anyone answer? Can anyone answer this? What are myotubes? If you've attended the lecture, I think you can answer it. What are myotubes? One, supercoiled structure. Okay. Only one person is answering. No one else. Myocytes align and join to form myotubes. Very good. Yeah, correct. Bundle of muscle fibers. Okay, okay, so what are myotubes? Myotubes is a developmental stage, right? After myocytes, not before myocytes. There only that statement becomes false. A developmental stage of a muscle fiber composed of a syncytium, like nuclei, formed by fusion of myoblasts, right? So the end product is a muscle fiber. But how is a muscle fiber formed? Like from where it starts? Within that procedure, there is a structure called as myotubes. People are still entering. I want everyone that please be on time. Okay, so this is the overview of the myotube formation, right? So here I have shown the picture till the formation of myotubes. After these myotubes are formed, they go on to become a bundle and form muscle fibers, right? Myofibers and muscle fibers. The first stage is the myoblasts, right? Myoblasts, the stem cells which differentiate to form myocytes, right? And these myocytes then migrate and adhere to each other and fuse to form myotubes, like in a longitudinal section you can say. They form myotubes. So the statement was multiple myotubes joined to form myocytes. No, multiple myocytes joined to form a myotube. The statement was exactly the opposite. Okay, so it was a wrong statement. So the next question is, which of these is non-striated? Skeletal muscle, cardiac muscle, smooth muscle, or rough muscle? Which of these is non-striated? Skeletal muscle, cardiac muscle, smooth muscle, or rough muscle? Okay, smooth muscles. Everyone is saying smooth muscles? Is there anyone who thinks it can be something else? Whoever is raising hand, please write your query in the chat box. I can't know if you just raise the hand, right? Which of these is non-striated? Okay. Okay, as everyone is saying, all of you are correct. The correct answer is smooth muscles. Smooth muscles is a type of non-striated muscles, whereas skeletal muscles and cardiac muscles are striated muscles, and there is nothing like rough muscles as we have discussed in the first question. So here I have already explained you this figure. What people are still coming? Okay. So first of all, cardiac muscles. Cardiac muscles are striated, branched, and uninucleated. They have only one nucleus, right? And these occur in the heart, as I've said previously. And these are involuntary muscles, right? These are involuntary means they are not in the control of, they are not in control of us, right? Heartbeat doesn't beat through our control, right? We can't control our heartbeat. So that is the nature of a involuntary muscle. And next is skeletal muscle. Skeletal muscles are striated as well, like the cardiac muscles. You can see the strips. From the structure itself, we can say that it is striated or non-striated. You can see from here that smooth muscles doesn't have striations, that is lines, strips. So these are smooth muscles are non-striated. So skeletal muscles are striated. They are tubular in nature, as you can see here, tubular structure, multinucleated fibers. So the main difference between cardiac muscles and skeletal muscles is cardiac muscles have only one nucleus, whereas skeletal muscles have multiple nucleus. Whereas both of them are striated, but there is a difference in the number of nucleus. And skeletal muscles, as the name says, they are attached to the skeleton and they are voluntary in nature. What is that? Voluntary is the movement is under our control. Skeletal muscles are present in our hand, legs, and we can move our hand and legs according to our wish. But for cardiac muscles, like in the heartbeat, it moves according to the process of the body, not because we want it, we don't want it. It doesn't work like that, right? And the last one is the smooth muscles. Smooth muscles are spindle shaped, as this structure is called spindle structure. As you can see from the figure here, these are spindle shaped and non-striated. You can see there are no striations, and these are as well uninucleated like cardiac muscles, and they occur in the walls of the internal organs, like we just discussed about the esophagus, right? Same way they are present in the stomach as well, and other internal organs, and they are involuntary in nature. Obviously, whatever movement is going on in our involuntary internal organs, we don't know that. We can't even feel that correctly, right? Nor we can control their movement. They are involuntary in nature. Okay. So we'll move on to the next question. Satellite cells are dash analog for skeletal muscle. Satellite cells are dash analog for skeletal muscles. Stem cell, kidney cell, myotube, or apoptosis. So satellite cells are what? In skeletal muscles. The question is asking that. Are they stem cells, kidney cells, myotube, or apoptosis? Start answering. Okay. Most of you are saying stem cells. Precursor of myocytes. It is not. Yeah, okay. I'm not asking for the explanation now. Just write down the answer. We'll discuss the explanation as well. Okay, most of you are saying stem cells. Okay, so the correct answer is stem cells. Good, good, good. So, first of all, I want to know what are satellite cells. Obviously, from the answer, you can know it is stem cells, but I want a correct definition. Does anyone know it? Like, can you explain satellite cells in a more elaborate way? What are satellite cells? Is anyone answering? Should I wait? They differentiate to myotube satellite cells or stem cells which differentiate to myocytes. Okay, good. Precursor cells. Okay, I understood that you all know. Okay, so satellite cells are obviously primary stem cells in the adult skeletal muscle, right? Not all muscle types have satellite cells. This is particularly for the skeletal muscles. They are these primary stem cells that form into skeletal muscle and are responsible for most of its functions like muscle growth, hypertrophy, regeneration. Stem cells are very important for regeneration. Since their original identification, satellite cells have been considered as unipotent myogenic precursor cells. So, in the previous class, I've already discussed what is pluripotent, what is multipotent, what is totipotent, what is unipotent. So, unipotent is that they have just a specific function. They are specific to a single function, right? That's why it is they are known as unipotent cells. Okay, if it is like for first of all is the totipotent cells like the zygote can differentiate into all kinds of cells in the body. After that comes the pluripotent cells, those are the embryonic stem cells, and then comes the multipotent cells, right? That is the hematopoietic stem cells and mesenchymal stem cells, and from those stem cells last one is the unipotent stem cells, and satellite cells are unipotent in nature. That is, they have the potency to differentiate only into a one single kind of cells, that is the skeletal muscle cells. Okay, so here you can see the embryonic progenitor cells are there, which give rise to all kinds of stem cells in our body. They give rise to satellite stem cells, committed satellite stem cells. These stem cells go on to become myoblasts, and myoblasts then differentiate to form myocytes, the cells, and which eventually forms the myotubes or myofibers. And after this, After myotubes, the bundle of myotubes are known as muscle fibers, right? Muscle fibers. I hope it is clear. We'll move on to the next question. Which of these are non-excitable cells, right? Which of these are non-excitable cells? Skeletal muscle cells, motor neuron, cortical neuron, or nephron? Your time starts now. Start answering. 63 students are here. Am I getting answers from every 63 of them? Hopefully, yes. Right? You need to be active in this session, right? Like, I'm not saying anyone to speak or show their faces. You just have to write down in the chat box whatever choice you are thinking is correct. That's a very easy thing. And yeah, it is very important to attend the lectures. The whole motive of this course is that you know animal physiology very clearly. Okay, so it is very important to attend the lectures and then practice the lecture through these questions. This interactive session is for your practice so that you can know that you are able to answer the questions or not after attending the lectures. If you're not able to answer the questions correctly after attending the lectures, you should go back and watch the lectures again, right? Okay, has everyone given the answer? Nephron? Nephron is your answer? Let's see if it's the correct answer. Yes, it is the correct answer. Nephrons are non-excitable cells. So, who can tell me what is a nephron? I guess everyone must be knowing it. Just write it down. What is a nephron? Where are nephrons present? Nephron is the unit of kidney. Okay. Only three answers? Okay, okay. Good, good. Everyone knows what is a nephron. Good. Okay. So, can anyone tell me what is the difference between excitable cells and non-excitable cells? Does everyone know what is the difference between excitable and non-excitable cells? Whoever are joining last moment, I want to request them all to please be on time. Already half of the session is finished based on the transmission of signal. Yamuna is saying. Okay. Excitable cells pass signals. Do non-excitable cells doesn't pass signals? See, they generate action potential. Okay. Those who are late to the class, I want you to please join in time. Already we have discussed 6 questions. Non-excitable cells don't generate action potential. Okay. Let's see. Okay, okay. Most of you are giving the right answer. So we'll discuss. So excitable cells have the resting potentials that range from minus 50 millivolts to minus 85 millivolts, while non-excitable cells have potentials that range from minus 5 millivolts to minus 10 millivolts. So excitable cells, like examples of excitable cells are neurons and skeletal muscle cells, whereas non-excitable cells can be red blood cells, kidney cells, etc. Okay, so so the main difference between it is not like that non-excitable cells doesn't have action potential at all, that they do not have the resting potential at all. They have, but it is very minimum, whereas excitable cells have a resting potential that range from minus 50 to minus 85. right? There is a more transport of ions throughout the membranes of these cells, right, throughout the membrane of these excitable cells, whereas the movement of Na plus and K plus ions, which generates the action potential in the cells, it is minimum in the non-excitable cells like the kidney cells, which are the nephrons. Okay, this was a very simple thing. We'll move on to the next question. Which of these has no actin? We are going deeper into the muscle physiology. Which of these has no actin? A zone, I band, H zone, or AI zone? Okay, start answering. Okay, H zone, you all are thinking? Okay, H zone, H zone, H zone. Okay, okay, let's see if it's the right answer. Yes, it is the right answer. H zone doesn't have any actin. So first of all, I want to know what is actin. What is actin? Where is it present? Actin is a protein, obviously, we can know it. Where is it present? I want to know from all of you. Obviously, it's present in muscles. I know that. I want to know the, yes, thin filaments present in muscle. That is the correct answer. In muscles, we know that, right? Okay, what is the other name of the protein which goes along with actin, which acts along with actin? I guess all of you know that as well. Yes, myosin. So myosin is present in thick filaments and actin is present in thin filaments of muscles, right? So this is the H zone. We were talking about H zone, I band, M line, right? These were the options. Go back and admit people. Okay. So here you can see these, the actin filaments are the blue colored structures and myosin filaments are the red colored structures, and you can see that in the H zone there are no actin filaments. Okay, this is the figure of a process of muscle contraction, like the thin and thick filaments slide above each other, and when the muscles are relaxed, as you can see here, these are relaxed, and when the muscles are contracted, right? So you can see that in the H zone there are no thin filaments, that is actin is not present, and whenever the there is a whenever the muscles are relaxed, the H zone is large, and whenever the muscles get contracted, the H zone decreases with the contraction of the thin and the thick filaments, as you can see here, and the middle line is called as the M line, whereas the I band is a mixture of the both of the myosin and uh, actin and the uh, myosin filaments, that is the myosin tail, myosin head and tail, you can see here, and this is the Z disc. Okay, okay, so we'll move on to the next question. Calcium binds to which of the following muscle? Calcium binds to which of the following in muscle? So these are the proteins present in the muscle, and you have to choose which of these is the protein to which calcium binds. Actin, myosin, tropomyosin, or troponin? Okay, troponin. Okay, most of you are writing troponin, and great job. Troponin is the right answer. So calcium binds to tropo troponin, not actin, not myosin, nor tropomyosin. So we'll know how and why. So you can see the structure here. Previously, the structure was of the same contraction of muscles. This is the same, but here you can see the troponin and tropomyosin involved in the muscle contraction, and you can see uh, the blue color structure is the troponin and the green colored structure is the tropomyosin. You can see how they are spirally coiled with the actin, right? That is the thin filament. The purple structure is the thin filament made up of actin units, and along that the tropomyosin and troponin are coiled together with these thin filaments, and the myosin heads come to bind to this thin filament. But how will it bind to it? It needs a site to bind, right? So which is the site? The site is present on the thin filament. So first of all, tropomyosin and troponin work together to block the myosin binding sites on actin. So troponin and tropomyosin cover these myosin binding sites which are present on the actin, so that myosin doesn't bind to it. Myosin doesn't bind, so movement is not there. But when these calcium ions come and bind to the troponin and tropomyosin complex moves from the place, and hence exposing the myosin binding sites on the actin, right? You can see the socket here. This socket is like to bind to the actin in the thin filament, but these are covered, these sites are covered by troponin and tropomyosin. But when calcium ions come and bind to the troponin, you can see here calcium ion binds to troponin, and as a result of which these troponin and myosin get together and move from their place and expose the myosin binding active sites, as a result of which the myosin head binds to the thin filament actin in the thin filament, which results in a muscle movement. Okay, so we'll move on to the next question. This is the last question. Large amounts of dash is present in smooth muscles in place of dash protein. Large amounts of dash is present in smooth muscles in place of dash protein. So what is the protein present in smooth muscles? D. D is tropomyosin troponin. Large amounts of dash is present in smooth muscles in place of dash. Read the question properly. It is written smooth muscles, right? Think properly and answer. Is it tropomyosin? Large amounts of tropomyosin are present in smooth muscles. Do you think that? Okay. Some of you are giving C, calmodulin and troponin. Calmodulin is present instead of troponin in smooth muscles, or tropomyosin is present in place of troponin. So are you all saying that in smooth muscles troponin is not present and tropomyosin is present? Whoever is giving the answer is D. I'm asking them. Okay, so the correct answer is C. C is the correct answer, not D. Right? Tropomyosin and troponin, both of them are present in skeletal muscles. But when it comes to smooth muscles, tropomyosin is present in smooth muscles. But instead of troponin, calmodulin is present. So the answer is large amounts of calmodulin is present in smooth muscles in place of troponin protein. In smooth muscles, troponin is not present, and instead of troponin, calmodulin is present. Okay. So whoever is given the answer is C. Good work. Good job. So we'll know. So what happens in the smooth muscles? Whenever there is an increase in the systolic calcium ions, right? Calcium, whenever there is an increase in the systolic calcium and ions go and bind to calmodulin. Previously we discussed that in skeletal muscles, calcium goes and bind to troponin, but here there is no troponin, so calcium goes and bind to calmodulin, which is present instead of troponin in smooth muscles. So as a result of this, calmodulin, which is in the inactive state, and after the calcium ions bind to calmodulin, it is becoming active, right? This results in activation of this calcium calmodulin complex results in the activation of the myosin kinase, right? This is the protein. With the activation of the myosin kinase, and phosphorylation of myosin light chain occurs. The myosin light chains get phosphorylated. You can see here these blue structures are the myosin filaments, and the myosin filaments get phosphorylated by kinase. Kinases itself, they have a function of phosphorylating proteins, right? So they phosphorylate the myosin light chain, and due to the phosphorylation of this myosin light chain, there is activation of the myosin ATPase. With activation of the myosin ATPase, myosin head gets attached to actin. So our whole motive was to attach the myosin to actin. Like we discussed previously, in skeletal muscles also, calcium ion binds to troponin, as a result of which troponin tropomyosin complex moves, exposing the myosin binding sites, and then the myosin head comes and binds to actin. This was the whole scenario in the skeletal muscles. Whereas in the smooth muscles, there is no troponin, calmodulin is present. So a different pathway goes here. So whenever there is an increase in the calcium ions come and bind to the calmodulin, which was inactive before, but now it has become active, and they form a complex called as the calcium calmodulin complex. Okay, and as a result of which they activate the myosin kinase, which phosphorylates the myosin light chain. And when these myosin light chains get phosphorylated, they release ATPase. They activate the ATPase enzyme, which helps in their movement and attachment to the actin, right? They help in their movement to the actin, and thus there is a contraction by the sliding filament mechanism. The mechanism is same. Skeletal muscles, so your smooth muscles, mechanism is same. That is the sliding filament mechanism. So this is the whole concept. Yeah, ninth question I've removed because that was not in accordance to whatever it was a out of box question. It was not included in the lectures. That's why I've removed the ninth question. Okay. Okay. So this is all for today. Okay, if you have keeping a record of your answers, correct answers and wrong answers, I want to know the score. Out of 9, how much you have got? 8 on 9, 7 on 9, 8, 7, 6, 8. Okay, I'm happy. Most of you have got 8, 6, 7. It's really nice. It's really nice. Keep learning, keep attending the lectures. Try to think properly and answer. Is there no one who has got full marks? 9 on 9? Is there no one? Okay, so 8 is the highest score. Is it? Okay. I wish in the next class we can get a 10 on 10. Today there were 9 questions. Last question got it wrong, ma'am. Okay, no problem. No problem. I guess you have understood the questions well. I guess you have understood the concept behind these questions. And if not, you can ask your queries in the discussion forum. I will be checking it continuously. If you have any doubts, ma'am, kindly send all questions. I will upload this PPT. I guess you all will be getting links for the PPT, and you can go through the PPT once again. Okay? And we'll meet again next Tuesday with a new set of questions from week 4. I request all of you to please attend the week 4 lectures and then come to the class. So maybe then you can get a 10 on 10, right? Okay. And whoever was new to the class today, who has joined today, I request them to please go back and listen to the lectures as well as these live assignment sessions. I have been recording them and uploading in my YouTube channel. I guess you have been getting the links of the YouTube channel as well. You can go through the live interactive sessions through the YouTube channel as well. Okay. I will be uploading this session on the YouTube as well. Okay. Okay, so we'll disperse now. We'll meet again next Tuesday. Keep learning. Thank you. Bye. Thank you. Thank you, all of you.